And he's also studied activity-dependent dynamics of spine stem resistance that could be used as a synaptic weight knob. Interestingly, these ideas I got to work on with Will Rawl in the late 1960s, my first foray into computational neuroscience. Today's talk by Tim will be in a different direction. Interesting, we're anxious to hear about this on the purpose and origin of spontaneous activity. Welcome, Tim.
Thank you very much. Thank you very much indeed. All right, I'm going to share my screen. And I suppose that'll mean that I can't see you guys anymore. I'm going to start with a very small plug that might be nice for those of us in lockdown. There haven't been any summer schools this year, but I wanted to take the opportunity to make you aware of a summer school that's happening in Cape Town every year. That's called the Simmons Ebro Computational Neuroscience in Ebro. It is by far the most diverse place you can ever possibly do neuroscience, in that we're collecting 20 students from the African continent and another 10 from the rest of the world. And it's quite a beautiful place to do computational neuroscience as well. You see here one of the views that you might enjoy if you were to apply successfully at the address down here, imbizo.africa. So I would greatly encourage those of you who haven't been to a summer school in the past to consider applying next year for 2022. It happens every year in the summer, so in the last 3 weeks of January. And if you're wondering, that's of course the Southern Hemisphere summer. With that, I'd like to start today's feature on the purpose and origin of spontaneous neural activity. And I should say right from the beginning that this work is the sole work of my postdoc Chaitanya Chintaluri, who you see here with a pizza that he has just baked during lockdown. So aside from being a creative scientist, he's also a creative and incredibly good cook. In fact, I still dream of the Indian food that he cooked me a few years ago when we went on a lab retreat. So if you should ever meet him, you should ask him to cook for you and not only explain the world of mitochondria. And then I wanted to also thank the organizers and thank specifically John for this invitation. It's actually the second time that I'm speaking at NYU, and the first time was 15 years ago when I was still a grad student. It was the first time that I ever got invited to give a talk. And it's quite a pleasure and an honor to get re-invited. I hope I'll do it justice. What you see here on the left side of the screen is actually my apartment in New York when I lived up on 151st and Broadway. This was the living room, and I'll come back to this apartment about halfway through the talk. I should only say that for those of you who live in New York City, it's probably not a totally uncommon occurrence that in the winters, your apartments get so hot that the only way to maintain a proper working temperature is to open the windows in those apartments and let the heat out from time to time. And that is a theme that will come back to us, as I said, throughout the talk. But before we go there, I'm talking about neuronal spontaneous activity, talking about spikes. So it might make sense to talk about what we think neurons fire to begin with for. Clearly, we think it's for them to communicate with each other, to make the brain do the amazing things that it does, to think, to compute, to walk, to talk. Simple models of that are models of propagation. So here's one on the left of the screen, a simple model of feed-forward propagation. And in fact, this very slide, it's actually not this totally very slide, but both the graphic elements on the right and on the left are graphic elements that I started my talk with 15 years ago. So I spoke then about modes of propagation along synfire chains, and it was then that Alex Reyes several times interrupted me about synchrony and asynchrony, and so I'm hoping that I might not get interrupted as much by Alex today, but he may remember this not as well as I do. I have it quite vividly in my mind. Both of these modes of propagation that I spoke about rely on something very important, which is noise. So no matter what kind of propagation mode you choose, whether it's synchronous, packets of spikes that travel along these layers of networks, or whether you think it's asynchronous propagation, where each neuron fires in an unreliable way, but as a population they still communicate correlatedly. Both of these types of propagation rely on additional noise that gets fed into these networks. And my PhD work in Larry Abbott's lab was focused very much on producing that kind of noise by way of self-sustained background activity in bigger networks into which these feed-forward chains would then be incorporated. And so you see the statistics of one of these networks here with a spike raster of 200 some cells firing away asynchronously with asynchronous and irregular statistics, balanced and with neurons that fire every now and then at an average rate of 10 hertz. To produce these networks, you had to search the parameter space of excitatory and inhibitory conductances here. Can you see my pointer, by the way? I'm not sure you can see my arrow. Okay, great. Quite extensively, because it turned out that this regime of self-sustained activity in which you have enough spikes at any given time to produce the same number of spikes in the next time step was not totally robust. So in orange here, you see the regime within which this network is self-sustained. But here you see that in that regime, the firing rates become quite high quite quickly. And in fact, the network starts to oscillate wildly. So you had to tune the network to stay here at this bottom edge of this self-sustained regime. And that meant that these networks died quite often spontaneously. So they fell silent after a moment of network-wide synchrony and then never came back to life. And that's, of course, something that we don't observe in nature, right? People don't generally fall over on the street and never return back to a healthy regime of neuronal activity. In fact, no matter where you look in the nervous system, you find that spontaneous activity is a quite common occurrence. So here on the right, you see a data slide that is probably quite familiar to you. It's from the famous Schultz, Dayan, and Montague paper about VTA, dopaminergic neurons. And usually people will focus on when the stimulus is delivered or when the reward is delivered. But there's another interesting part in this plot in that before any stimulus is ever delivered, these neurons fire quite actively. So there's a lot of spontaneous activity. Here, it's also seen in olfactory neurons, in neurocortical pyramidal neurons, in Purkinje neurons. Anywhere you look, you'll find spontaneous activity at various firing rates. So that seems peculiar from an energetic point of view, right? Because spikes cost money, and so there must be a function to spending that money. Why would the nervous system not save the cash at moments when it really doesn't need any spikes and when spikes are presumably only serving to communicate? What is it that they're communicating when there's nothing to talk about? And when I talk about money, I of course mean ATP. So adenosine triphosphate is the fuel that makes everything run in your cells. That is the energy that's being used. It's made from ADP, so ATP is turned into ADP, ADP is turned into ATP, and vice versa, either by adding a phosphate or losing it, and that is an operation that creates, when you go from ATP to ADP, energy that you can then use to drive, for example, ionic pumps that will pay for the spikes. And that's not a totally negligible bill. it's 15% of the energy budget, for example, for mossy fiber neurons, it's estimated. So if that is the case, why fire in the absence of sensory stimuli? Okay, so of course, the energy that's being put into ADP to make ATP is a little bit more complicated. The pathway is a little bit tricky. I have simplified it here, and I'll take you through what's happening. Energy is usually what we consider glucose or lactate that gets turned into pyruvate, shuttled into the neuron, and then delivered to the mitochondria. And in the mitochondria, it's used in the Krebs cycle, in the citric acid cycle, to produce NADH, which is then used to pump protons across the inner membrane of the mitochondria from the inside of the mitochondria. And then this gradient of protons between the intermembrane space of the mitochondria and the inside is used by ATP mills that are essentially milling ADP into ATP by allowing protons to return back into the mitochondria. That process is then repeated multiple times as ADP is recycled and ATP is used and converted back into ADP. It's important to note here that for neurons, the rate-limiting element in this process is not the pyruvate that's provided to the mitochondria, but rather it's delta psi, so it's the gradients of protons between the inside and the inner intermembrane space, as well as ADP. So ATP and ADP are limited resources in mitochondria, and when there is no ADP around, then this process is going to come to a standstill. But pyruvate here is delivered at a relatively constant rate. Okay, so we're going to drill open the electron transport chain a little bit more. I've expanded it here, and I told you already, NADH is being burned into NAD in order to pump protons across the inner membrane of the mitochondria. That process allows not only protons to be pumped up, but also demands that electrons are being passed onto the electron transport chain. This is actually a process of multiple cross-membrane proteins on the inside of the mitochondria that will pass these free electrons from one complex to the next, eventually turning these electrons together with protons and oxygen into water at the end of the electron transport chain. It's also worth noting that this is not what happens to every electron on this electron transport chain. In fact, some of these electrons will hop off of the electron transport chain, they will escape, and they'll recombine with free oxygens and become what is called radical or reactive oxygen species. And so these are relatively harmful substances. Think of hydrogen peroxide, think of drinking that. Then you know how the cell feels about reactive oxygen, probably in a close approximation anyway. And there are various scavengers at work inside of the cell that are making sure that there's not too much reactive oxygen around. So pumping H+, pumping protons across the membrane of a mitochondria routinely produces this reactive oxygen. And so we can plot the amount of reactive oxygen in a cell as a function of the ATP consumption. The more ATP is being consumed, the more needs to be produced. And so Ross concentration will go up with ATP consumption. This is a known fact. So this is not our work, obviously. If any of you know me, I'm by no means a biochemist. But the important thing here to take home is that too much spiking is bad for the cell, or strictly speaking, too much ATP consumption is bad for the cell because it will produce as a side product these free oxygen or these radical oxygen species. All right. Now, I already told you that ATP and ADP are limited resources, so it's also possible that this electron transport chain will come to stall by the fact that there is no ADP around. So when there is no ADP around, we can't use these protons to mill ATP because they're all milled. And when that happens, you can imagine that the proton gradient on the in the intermembrane space of the mitochondria will quickly escalate, and so will the electron transport chain, the electrons on the electron transport chain themselves. So before, when we had to produce a lot of ATP, you could imagine that this was a situation in which there was a lot of traffic, and with increased traffic, there was more reactive oxygen that could hop off of the electron transport chain. Now we have a situation in which there is a traffic jam, because it turns out that in order to get rid of these free electrons in the electron transport chain and make them into water with oxygen and hydrogen, obviously, hydrogen needs to also be pumped across the membrane. So when this transport chain starts stalling, there is something that happens that's called reverse electron transport chain, ROS production. So electrons that are stalling, sitting on the transport chain without being passed on, will also hop off, escape into the cellular or the mitochondrial space and produce more reactive oxygen. Okay, so too little spiking is also bad for the cell. That means that this function here is not monotonous, but actually has a sweet spot down here, a non-monotonic shape otherwise, such that if you're below a certain baseline of ATP consumption, you will produce more reactive oxygen and is good for you, and above it too. Okay, these two different kinds of reactive oxygen are referred to as reverse and forward electron transport ROS, reactive oxygen species. And again, this is published stuff. All right. So again, I want to drive home the point here that it's not pyruvate, so it's not the energy fuel that is delivered to the Krebs cycle here that limits the rate at which ATP is produced, but it's actually ADP and delta psi. Okay, so that means that this Krebs cycle here that's producing NADH to drive the electron transport chain is cranking on regardless of whether this is stalled or not, and so producing more and more ROS when this is stalled. Oh. That means here that we can sort of create a respiratory state space in which we plot these two important parameters, the ATP concentration on the inside of the mitochondria on the x-axis, and the delta psi, the proton gradient on the y-axis, and we can plot sort of the amount of ROS as a heat map. So red is worse, white is good, and in this situation where you have little ATP around or a low proton gradient, you produce a lot of forward electron transport chain ROS. And in the situation that the electron transport chain is stalled, you produce a lot of reverse electron transport chain ROS. So there's two corners here in this respiratory state space that are unhealthy, namely the one where there's a lot of ATP around with a high proton gradient, or where there is little ATP around with a low proton gradient. All right, so that's the basic idea. It is that if a neuron consumes too much or too little ATP, it will potentially poison itself with reactive oxygen. And when a neuron experiences a sudden or periodic loss of inputs, its ATP consumption will decrease dramatically. So that's not only for the loss of output spikes, but of course also for the loss of ion flux due to postsynaptic potentials, which amount to up to 70% of the metabolic budget of any given neuron. So actually, when a neuron stops getting inputs, it seems like it's going to have a lot of ATP that it doesn't know what to do with relatively quickly. That's because in the absence of a regulatory mechanism, the Krebs cycle turns on, and so the neuron must be interested in dissipating energy. And that's, coming back to the beginning of my talk, very much like you sitting in a New York apartment in the winter, not knowing how to downregulate your heater. And in the absence of having a control knob for your heater, you open the window. And for the neuron, we postulate that that's firing an action potential. So the idea here is that spontaneous firing is actually triggered by intracellular reverse electron transport chain created ROS, and serves to relieve ROS poisoning by dissipating or wasting energy. All right? So, the logic of this hypothesis, we wanted to confirm in a model. And so for that, we built a vanilla integrating fire neuron. And then I'm going to show you that we're going to add a mitochondrial module to model a metabolism for ATP, and then additionally add a ROS module that simulates the creation of reactive oxygen species in the mitochondria. And then finally, we'll add additionally ion channels that detect ROS and see hopefully if this model makes sense, and on the way also check with the literature as to whether there is reasons to doubt the story I'm telling you. All right, so we're starting with a mitochondrial model, and mitochondrial model actually is to be had already published by Christine Nazareth in 2009 in the Journal of Theoretical Biology, where she and her colleagues built a simplified model of mitochondrial ATP production. It has the elements we need, so it has a pyruvate intake to the mitochondria, it has a proton gradient buildup here, it has ADP to ATP conversion, and then that ATP gets shuttled out of the mitochondria to be used up by other cellular processes and turned into ADP, which is in turn shuttled back into the mitochondria, and there it will be turned again back into ATP using the existing proton gradient delta psi. That also means that in Christine Nazareth's model, that ATP consumption outside of the mitochondria dictates pretty much everything else. It dictates specifically psi and ADP to ATP conversion rates in the model. So you see this here in this rate, in this plot where I'm plotting on the x-axis ATP consumption, and on the y-axis in arbitrary units, the concentration of mitochondrial ATP, as well as the proton gradient between the inside and the intermembrane space of the mitochondria. The other variables of this model also follow from this interaction or from the ATP consumption. So again, ATP consumption here is on the x-axis. On the y-axis, we have both the concentration of NAD, mitochondria NAD, as well as pyruvate in the mitochondria. And finally, also all the other reaction rates of the different processes going on are dictated by ATP consumption outside of the cell. These latter plots are not so important, but the first plot I showed you was ATP consumption against ATP concentration inside the mitochondria, as well as delta psi, is what we've already encountered once when I was telling you about the respiratory state space, and we can replot that here with delta psi on the y-axis and ATP, mitochondrial ATP on the x-axis. And now we can essentially know exactly where any given neuron is if we know its steady state ATP consumption. So if you have the resting metabolism of a neuron, that's say, for example, at 20 here, effective rate constant 20, or if you want ATP molecules used per millisecond, then you know that it will sit at the intersection here of 0.7 delta psi and 0.9 ATP molecules, let's say, or concentration internally in the mitochondria. Okay. Remember also that I've told you about ROS being built up as a function of the ATP consumption. So before I only drew this as a sketch, here we're now making this a little bit more formal. We're taking the experimental evidence that we've seen before that showed both ret and fet ROS production, and we're postulating a formula that we can then plug into this model to add an additional facet, which is the concentration of ROS inside the cell as a function of ATP consumption. Okay, so we can now add this to this respiratory state space as we've done before, because we know, again, ATP consumption is at a certain value. And for that value, as well as for all other ATP consumption, we can now create a heat map. And I apologize that I haven't put the color bar here. This has slipped my mind again and again in preparation for this talk. But we now know not only how delta psi and ATP, mitochondrial ATP concentrations are related to one another, but we also know how much ROS we're expecting inside the cell for a given combination of these two. And of course, this is not only true for one neuron, but also maybe for another one that has a higher resting state, so it naturally uses more ATP in a given time interval, and that also means that it has a different amount of ROS that's being produced, as is indicated here in this triumvirate of variables for the mitochondrial ATP, for the proton gradient across the mitochondrial inner membrane, as well as the ROS concentration. Okay, now notice how neither of these two neurons, or by the way, this could also be a single neuron in two different states, once receiving inputs and once not receiving inputs. But notice how neither of them are at the sweet spot. They could be better at not producing ROS if they changed their ATP consumption. And so that's the whole idea here, right? That this neuron would go and look at its ROS production and say, well, you know, I really ought to be using more ATP to make my way to this minimum ROS production here. And so if I only were to spike once, as shown here in the upper right, where I'm plotting the membrane potential of this neuron on the y-axis and time on the x-axis, you see that in the parallel plot below it, where I'm plotting ATP consumption on the y-axis, and again, time on the x-axis, you see how the ATP consumption, the energy consumption we postulate, is going up by a certain amount, a cost that is associated with re-equilibrating the cell after the occurrence of an action potential, and then it will fall back to its steady state ATP consumption after that spike eventually. So in the respiratory state space, this means that this neuron will take a trip away from the position it's in. It will reduce its proton gradient as it's using that proton gradient to rebuild its ATP stock. And initially, it will also reduce its ATP stock as that's being used up to re-equilibrate the neuron. And then as the mitochondria catch up, the neuron will eventually return to its original baseline state of ATP consumption into this less healthy regime of ROS concentrations, as indicated here in the red color on this heat map. Okay,
Tim?
Yes.
Tim, we have a question here, accumulating votes,
Sure.
by Rob Fromke, who would like to come on screen to ask you the question.
Let's do it. He's not coming online, is it?
Sorry, just a minute with this.
Waiting. I think you're on, Rob. Oh, we don't hear you. You need to unmute.
Hey, Rob. How you doing? I like it when you can't ask me the question.
You
You
hear
can't
me?
hear me?
Vishwa, can you make sure he's unmuted? And
If you float your mouse over the
Yeah,
screen,
Rob is unmuted. I'm not sure if something's off at his end.
What about now? Your audio
Yes,
just came back
that's
up.
working.
Good.
Okay, great. Yeah, thanks, Tim. This is super interesting. I didn't know you'd be going in this direction. Maybe before you get too much further along, can you just give us a little bit of a sense of the biology of the reactive oxygen species? Kind of over what time scale do neurons have to control this before things get out of whack and it's really going to do damage to the cell? Are we talking minutes? Are we talking hours?
So I think
What
this is a
happens?
matter of scale, right? So if it's just a little bit, there's probably a relatively good tolerance for a deviation from the optimal Ross, as there is scavengers in place that will grab free electrons or that will grab Ross to eliminate the threat. But if you have extended periods of high firing, I would estimate that it gets hot pretty quickly. So I can't tell you exactly what the timescales are. Again, I think this is a matter of whether we're talking about a little bit, then there's a trade-off. If you have massive overproduction of Ross in short timescales, then you want to get rid of it pretty quickly. But it might also be that you can tolerate small deviations or smallish deviations for a long period of time. I should also say that this plot that we're driving here, this V-shaped plot, is on a log-log scale. And it also doesn't have to necessarily be the plot, right? This is what we're postulating from squinting our eyes and looking at the experimental literature. And so it's not clear that it's going to go up quite as dramatically as I'm plotting it here, as Chaitanya is plotting it.
Cool, thank you. You can turn me off.
All right, and I'll go back to screen sharing. All right. We were here where a neuron took a quick trip to a better Ross environment and then eventually returned to its ATP baseline consumption state. You can imagine that neurons can have very different baseline consumption of ATP, all of which are going to lie along this trajectory in the respiratory state space, or in other words, along this V-shaped function here when it's plotted against ATP consumption alone. All right. So this is so far the logic of producing Ross and then also avoiding Ross by firing a spike. But in order to implement this in a cellular model, we would obviously need a mechanism to manipulate the membrane potential based on ATP consumption or Ross concentration per se. So in other words, we need ion channels that would respond either to FET or to red Ross. Okay, let's start with forward electron transport chain Ross. So the electrons, the free electrons that routinely hop off of the electron transport chain when there is a lot of traffic. In other words, when there's a lot of ATP consumption and thus a lot of ATP production. These could be channels like a ATP-sensitive potassium channel that is activated, that opens when there's little ATP around in the neuron, or a KCA channel that is activated during excessive calcium influx. Both of these channels would thus hyperpolarize the neuron and stop it from firing, stop it from consuming energy, slowing down the traffic along the electron transport chain, and thus reduce the amount of oxidative stress by the production of Ross in the neuron, bringing it further towards this sweet spot down here. Now, ion channel models for red Ross avoidance are maybe a little bit more complicated, and some of this are things that we're looking into right now. Some of this is well known. Make up your own mind, but the simplest explanation for avoiding red Ross by lack of activity or a stalling electron transport chain, would be an ion channel that contributes to a depolarization in the absence of activity. In other words, a persistent sodium channel, for example, that responds to, let's say, hydrogen peroxide, or a T-type calcium current that changes with a reduced cytosol. And both of these options are real. So there are persistent sodium channels and calcium channels like that will sense basically byproducts of increased ROS production, like, for example, a reduced cytosol when all the scavengers are being busy and at work and maxed out in their attempt to scavenge reactive oxygen from the cytosol of the cell. So these would be channels that simply open and then bring the neuron to spike and thus restart the electron transport chain when ATP is being milled from the now available ADP. Another possibility would be here adaptation, or the taking away of adaptation, production of additional spikes in a stimulated neuron by changing the inactivation profile, for example, of an A-type potassium or a calcium-sensitive potassium channel by way of NADPH detection. So again, this is a set of channels that are known to exist in the literature that haven't necessarily been thought of as promoting activity for a quiescent neuron. Here's another one. This would be, again, the T-type calcium, which would, in the face of ongoing activity, add additional spikes and convert a single spiking neuron into a neuron that is bursting and thus producing more spikes, which then would contribute to using more ATP, which would drive the neuron away from this more toxic regime of ROS production. And then finally, a more speculative one, but nevertheless maybe worth mentioning, reduction or even removal of a refractory period by manipulating resurgent sodium channels. For example, and here I'm a little bit out of my depth, I have to warn you, but I've been told that ROS increases may contribute to expression of beta subunits in these sodium channels, which would then lead to a much reduced refractory period for them, allowing for neurons to fire much more tightly spaced action potentials. So all four of these options, increased depolarizing conductances, loss of inactivation, activation of calcium through the redox state and creating bursters, or reduction of the refractory period, are ways that a neuron could stimulate or initiate a higher energy consumption. And for the next few slides, I'm only going to focus on one of them. So through increased sodium conductance, we're going to be able to manipulate ATP consumption by way of spiking and thus manipulate our position or a neuron's position inside this respiratory state space. But I wanted to quickly mention that all four of these variables are already represented in a relatively widely used neuron model, the adaptive exponential integrate and fire model, that is already been shown to produce quite realistic dynamics. And so we're now in the process of foregoing the addition of specific ion channels and expressing this all within a model in which the concentration of free radical oxygen species is linked to different parameters of this ADEX leaky integrate and fire neuron. And you'll have to hold off a little until we can show you the proper figures.
Tim, maybe you want to answer another question or two before you jump into that? Yuri Buzaki asks about spiking during up and down states of non-REM as a state when ADP balance is achieved. Do spikes during other spiking states of the neuron perform the same function, or you think up and down are different?
Let's come back to that question in a moment, because I'm going to go through a couple of examples here that might answer that question. And then if I haven't, maybe stop me again.
Okay.
Okay?
All right.
All right. All right. So here is a first example of what happens to a neuron in which we have a mitochondrial module with a ROS producing element, as well as a depolarizing ion channel. So this neuron would be naturally quiet because it doesn't receive any input currents, but because of its baseline ATP production and thus its baseline ROS production, the neuron will start depolarizing and thus start firing a spike. And as a result of that, both the ATP, plotted here in green, as well as the delta psi, the proton gradient, will dip together with the ROS concentration, which then will recover to a point where it becomes no longer tolerable for the ion channels that we've implemented here and drive this neuron to another spike. So this is a specific implementation of this neuron. So we've set the ATP baseline consumption to 40 here and the cost of a spike to 20 ATP molecules. We can also choose a different parameter set. So we could, for example, reduce the ATP consumption to 30 in this case. And what that means is that beforehand, we were sitting somewhere here at 40. This neuron sat at a baseline consumption of 40 and then jumped by way of spike initiation to the bottom of this pit here, and then slowly climbed back out until it again jumped back down. So it was repetitively taking this trip into the bottom of the well here. And in the case on the right, this neuron will much more quickly go up this steep hill here because the baseline consumption is less, so that it will much more quickly, rapidly start returning to regimes where ROS is harmful, and thus, will initiate another spike. And so you will see that this neuron spikes thanks to its different baseline consumption at a higher rate to maintain a more healthy regime of ROS. Okay, so here you have roughly the same average ROS production, but you have to fire at higher rates for it. And then we can consider another case in which we've massively decreased the cost of a given spike. So here, a spike will only cost you 1 ATP instead of 20 before. So you can imagine that this neuron will have to take many little jumps down this pit rather than one big jump. And that means it will have to initiate many action potentials until it's in a regime that is more ROS friendly. And this means in this case here that this neuron will fire in bursts, then climb back up the hill and burst again. And depending on these, the baseline ATP consumption, the shape of this bursting will be either a regular burster or an irregular burster. So maybe regarding that question that just came, the shape of the or the behavior, the firing rate behavior, the spiking behavior of the neuron is very much determined by where that neuron sits in terms of its steady state ATP consumption, but also how much it spends on a single spike. So this depends, of course, on morphology of the neuron. It depends on the overall, the length of the axon, the length of the dendrites, how much membrane it has to depolarize for a given action potential, and so on. So these are obviously just model examples, but we're in the process of fitting these more closely to different neuron types. Okay, so this what I've just said is sort of also echoed in these summary plots here, where I'm plotting the ATP consumption, the baseline consumption of the neuron on the x-axis, and the cost per spike on the y-axis. And you see that there's different regimes in which you have regular spikers for expensive spikes, where the neuron only has to fire a spike every once in a while, and bursting neurons for cheaper spikes, going all the way to quasi-epileptic or continuously firing, high firing rate firing neurons at the lower end when the spikes simply get too cheap or when ATP consumption is relatively low. And the same holds true, the same logic holds true for the other three plots in terms of their behavior. That's not as clear to see. Sorry, I need a glass of water here. And this is dropping. All right.
So, Tim, just let me mention there's about 10 minutes left.
Yep, we're almost done.
Okay.
All right, so quick summary. We have shown in a model that spiking may function as a protective mechanism against ROS toxicity. So kind of the other side of the coin of anti-excitotoxicity mechanisms. In our model, the action potential serves as a safety valve first, before it serves as a communication device. So this might make for a nice contrast to this idea of efficient coding where neurons really spike to talk to each other. So we postulate or we think that spikes may have evolved as a counter mechanism to the fact that neurons cannot control their pyruvate intake. So I've already mentioned this a couple of times. Neurons are not the masters of their own energy intake. Pyruvate gets shuttled in by the astrocytes. They generally don't do glycolysis to produce ATP very much. There is glycolysis in neurons, of course, but they don't have energy storage. They don't have buffer systems. They are driving their Krebs cycle at a relatively steady state. And so in the face of this ongoing energy supply, these what was to become neurons may have evolved mechanisms to get rid of that energy, so to not poison themselves. So here, ROS homeostasis is obviously an interesting additional mechanism, or obviously additional, maybe not interesting, but a mechanism that complements synaptic plasticity because it regulates the excitability of a neuron that is away from synaptic plasticity. So it's a homeostatic mechanism that is unrelated or not immediately related to what the synapses are doing directly. And so this could also present a fundamental constraint for wiring and coding in neuronal nets and may have a footprint in disease, obviously. And this is something that I would like to quickly revisit. At the beginning of the talk, I showed you a dopaminergic neuron that was firing at a relatively constant rate, and then it was elevating its rate in the presence of stimuli or decreasing its spontaneous rate in the absence of a stimuli or a predicted reward. And you also know that dopamine neurons are relatively important for movement initiation. So we thought that it might be an interesting place to look at for what happens when this goes, when ROS maintenance goes wrong. So the obvious place to look here would be Parkinson's disease. Parkinson's disease is a disease in which otherwise spontaneously discharging neurons in the substantia nigra cease to fire, and so they stop providing dopamine to other critical brain regions further up. So you see here, substantia nigra is the origin of this beautiful dopaminergic neuron, which then has an axon going all the way to up to here, where it provides dopamine by way of this rich and bushy axon terminal. And so a healthy dopaminergic neuron will have a firing profile that looks pretty much like this, where I'm plotting the membrane potential on the y-axis and time on the x-axis. When a stimulus is provided, it will respond with firing, but even in the absence of stimuli, you have spontaneous firing rate. And of course, for us, this looks very much like the baseline ATP consumption of this neuron sits somewhere here, such that it will need to provide additional spikes to sit down here in this optimality valley for ROS production. And so you could imagine that if something goes wrong with either ROS detection or maybe something goes wrong in electron transport chain so that there is no more ROS being produced thanks to whatever problem, which would then obviously also have downstream effects on the energy on ATP providing of the ability of the mitochondria. You could imagine that this neuron, which before had a strong motivation to travel to the valley of this V-shaped function, no longer has that motivation, so that it will no longer produce spontaneous activity because its ROS concentrations at the baseline firing rate are as or are thought to be, if the detector is broken, or are in reality quite as high as they were before in the optimal case. And a neuron like that will then no longer spontaneously fire and only respond to stimuli. Okay, so this was sort of an alternative idea to some of the origins of Parkinson's disease, and obviously we're taking our mouth quite full here, because I'm sure there's many more people that know a lot more about PD than at least me. Chaitanya has been reading up on this for a long time, and he found that there is a insecticide that's called Rotanone that produces pretty much the same symptoms as Parkinson's disease, and that conveniently actually is known to be a competitive agonist of a binding site in the complex 1 of the electron transport chain, so precisely where red ROS is produced most. So this insecticide will disturb the electron transport chain to make it impossible for it to produce ROS, or so we speculate. And that would, of course, go very much in line with this hypothesis of this broken second arm here of this V-shaped curve, and be sort of in line with the postulated origin of Parkinson's disease as proposed in this model. And then finally, to finish up, we also wanted to quickly look at how this works in neural networks. So we put together a bunch of these neurons that Chaitanya has created with simple integrated fire neurons, again with a mitochondrial module, and looked at what the activity was that they were producing. So remember that I started my talk with this image that I once 15 years ago introduced at NYU before. This is a network that produces spontaneous and self-sustained activity by way of being a critical process in that, in that it produces as many spikes in any given unit of time as are necessary to remain alive. And of course, I told you that these networks often died so that they spontaneously stopped producing this. This is one of the problematic features of a critical branching process, that sometimes they become subcritical or sometimes supercritical and they stop. When we introduce a mitochondrial module into these kind of networks, that no longer happens. You will see the network pause from time to time, but you see that it produces very much the exact same dynamics, or statistics at least, of the recurrent network without it, with the exception that it will no longer allow the network to die, simply because as it falls quiescent, ROS concentrations go up and neurons will spontaneously start to spike. So there is an additional homeostatic buffer in here that produces asynchronous, irregular, self-sustained activity. And we thought it was curious that the length of the activity bouts that we saw in these networks were on a power law quite similar to the ones that have been observed in the avalanche corner of the field. So we thought this may be what's going on in these cultures, or also in slices in other networks that are otherwise broken, that are not represented in their full glory in terms of connectivity and contextual environment, so that you get these neurons that are starting to fire spontaneously, that are rising towards a threshold, and that will gladly fire when they have enough inputs, but because the network is otherwise broken, they will stop firing again, or come to pause or stutter, quite like these neural networks that we have here with these ROS avoiding mechanisms. So with that, I'm coming to an end. I have a very easy takeaway message that maybe back in the day, way back in the day, neurons started firing spikes as a means to survive, so that they simply avoided ROS poisoning themselves, thanks to the inability to provide their own glycolysis, or means to regulate their energy intake, and that their screams, their action potentials, turn out to be useful for neural conversations, and that is the reason that since then neurons have been screaming at each other, and very gladly and successfully so. And so with that, I come to an end. I should mention all the people that have contributed to this work. Most importantly, of course, Chaitanya, who's also in the comment section I saw when I quickly went to answer questions, but also people in the Vogels lab more generally speaking. So here are some of them on a field trip, as well as people in the CNCB. There's a B missing here in Oxford, most prominently Anissa Kempf and Gero Miesenberg, who inspired many of the ideas that Chaitanya and I put forward here today, as well as Peter Hasenhüttl. And then there is the people that have written the papers that have inspired us as well. So Christine Nazaret and her colleagues, Anissa Kempf again, as well as Ina Slutsky and her colleagues. There's a long line of contributors on this paper. These 3 papers are really well worth reading. With that, I should also thank my funders, especially the Wellcome Trust and the ERC, as well as the BBSRC that has been funding Chaitanya's salary, and my new employer, IST Austria, who will, hopefully continue to provide great grounds to work on these ideas. And with that, I just want to finish saying that my lab has positions for postdocs, and that the IST has a great graduate school. So if you know people looking for a graduate school in interdisciplinary, not just neuroscience, and interdisciplinary sciences, send them to the vogelslab.org website or directly to the website of IST Austria. So I'll take questions. Thank you very much.
Thanks very much, Tim. That was very, very interesting. A new set of things for us to think about. We have quite a few questions here. Pierre asked a couple of questions. I can convey them to you. Do simpler brains, fly, C. elegans, also display spontaneous activity?
I don't know. I mean, flies have action potentials. C. elegans is a little bit different in that C. elegans doesn't fire action potentials at all. And in fact, it also doesn't sleep, or it sleeps differently. It doesn't have the same features that systems with action potentials display during sleep. We're looking into these things now. For fly, I would say definitely there is spontaneous activity, and in fact, the whole idea of Ross regulation came from a collaboration, an initial collaboration about something else with Gero Miesenberg and Anissa.
Okay, so Pierre also asks, how is spontaneous activity defined? Is it from the point of view of the scientist or from the point of view of the organism?
Yeah, I agree with that totally. So spontaneous activity obviously is termed from the view of the scientist, in that, it's spontaneous as it occurs seemingly without provocation. For the organism or for the neuron, it's obviously not spontaneous, in that it's at least according to the theory or the idea that we're putting forward, it's not just something that it's doing out of having a good day, but rather because it's trying to prevent the buildup of Ross. And so it's actually a purposeful, non-spontaneous, well-guided behavior.
Okay, perfect. So Badr has a couple of questions, and he wants to come on screen. So Vishwa, can you help that out?
Yep, he's coming on. Sorry, John, this might be taking some time. Perhaps you could just, for sake of time, maybe you could just ask this question for him.
It's a pretty long question. Let's see. Other cell types survive without having the ability to spike at all. In this picture, how do they manage to stay out of the dangerous regime?
So that's true. What's important to remember here is that other cell types have a much more tight control of their glycolysis and their energy intake. So what makes neurons really special here is that they don't do that. That in our sort of imagination, at least, what happened was that some neuron found itself, or some cell found itself in the unfortunate position of not being able to do glycolysis very effectively anymore, and then having a friend, an astrocyte, come to its assistance by sending loads of pyruvate over to that specific cell, which then created another issue for that cell in that the astrocyte kept sending pyruvate regardless of whether that cell needed the energy or not. And so at that point, the neuron had to become a neuron by firing action potentials to get rid of it. So I would say this is a necessity of not being able to control your energy intake. One could also argue that being a neuron is a little bit like a bank robber, in that if you're a bank robber, it's better to have your car running at all times so that when you come actually running out of the bank, or when the cops show up, that you can get away neatly. So for a neuron, it's quite important to be ready to fire at any given time, so that you keep a steady supply of ATP around for that purpose. And that would require that even, that given that this ATP is constantly being produced, you have to find a way to get rid of it every now and then.
Okay.
There is some sort of co-evolutionary thing going on here, at least in our minds, that's obviously not easy to disentangle. And the question here is also, what was the first action potential? When was the first action potential? And did that happen because of the pyruvate problem, or was it the other way around? I don't know. I mean, we're only starting to look to talk to people that know about these things in terms of evolutionary intricacy.
Sylvia raises the point, electrophysiologists sometimes see neurons suddenly start to spike, although they were completely silent for many minutes before, even when using dense electrodes to include artifacts from drift. Do these neurons work differently than described here, or does it have to be a recording artifact?
I don't see why they would have to record differently. I think, I don't, so again, I'm going to try to explain this within the framework that we're coming up with. That doesn't have to, there's maybe different ways to explain this as well. Within the framework that we've come up with, it would be that this neuron was quiet for quite some time and silently building up reactive oxygen. For a little while, there was compensatory mechanisms at play, for example, these scavengers that reduce the cytoplasm and keep Ross at bay, or you can also imagine other mechanisms at play that would similarly prevent further Ross production. But then at some point, this neuron couldn't hold out any longer and found that it had to depolarize its membrane potential to simply get rid of some of the excess ATP or provide new ADP to keep the electron transport chain going in the mitochondria. Whether that's really indeed what happened in the slices, who am I to say? That's what I would dream up as an explanation to remain within the framework of our idea.
I'll just take a moment here and ask a question about, some cells have their mitochondria go through oscillatory behavior on the timescale of minutes or even longer. Could that lead to ways of interacting with neural electrical activity?
I didn't understand the first part of the question. Some neurons, say that again?
Some cells, like neurosecretory cells, their mitochondria show glycolytic oscillations. Okay, does that have any impact in what some of the things you're asking about or studying?
I would raise my hands in the air and say I have absolutely no idea. I have never thought about it. I don't know.
Okay, I don't know if we ever got Badr on the screen here. Yu Chung asks, same question. Sorry, missed it. How would a cell differentiate between forward Ross and reverse Ross? Because if it is that they cannot, then by this model, an increase in forward Ross would lead to an overexcitation of the cell.
So it's essentially in the byproduct, right? That forward ROS comes along with an ATP. Like if you remember in the talk, I showed that forward ROS came along with not enough ATP in the mitochondria, and in the cytosol as well, so that it was actually regulated indirectly by a potassium channel that would open in the absence of ATP. And so fat ROS wouldn't be directly regulated by the same mechanism that red ROS is regulated.
So another question, we should have maybe one or two more questions. Very intriguing, Tim. Do you think that your rather regular firing patterns will be made realistically more random by spatial non-uniformity of ATP, multiple mitochondria, local regulation of ion channels?
So if I understand the question correctly, this is about the distribution of mitochondria within the cell. And so what we've talked about so far was, of course, the mitochondria mostly at the soma. One could also dream up a situation in which mitochondria at the axon terminals are experiencing a overflow in ATP or a lack of ADP, so to create other phenomena. I think that the heterogeneity of the firing patterns would be affected by that clearly, but I don't think it would be a small effect.
Okay, last question. This is from Annalise. What is the relevance of subthreshold spontaneous activity in terms of ATP consumption and ROS generation?
So subthreshold being simply postsynaptic potentials that are coming in, but that are not strong enough to evoke action potentials. So they are eating up a large portion of the energy budget that a cell has at its disposal. Some papers guess that it's up to 70% of a ATP production is going towards PSP re-equilibrylation, you know what I mean? Sorry. So it would take, a cell would take a large hit in terms of its ADP contingency if a neuron were to receive much less or much fewer PSPs than it had budgeted with. So that a neuron that lives in an organism that falls asleep, or a neuron that lives in an organism that experiences a monocular deprivation experiment would come to the conclusion that it would have to initiate evasive action in terms of firing action potentials in order to get rid of the ATP that is overflowing from the lack of inputs. Does that make sense?
Tim, thanks again. This was a great talk, and I know you're working very hard today, and hope you have a good weekend for that. And we wish everybody else a great weekend coming up. It was good to see everybody online, and we hope to see you again at one of these Swartz seminars.
It's been lovely. Thank you very much for the invitation.
Sure.
And thank you for the